So we're in your chart, in your notes. This top box, we're going to go ahead and write the word prophase. This is the first phase of mitosis. We had a box that said interphase. That interphase box on the other page of your notes, interphase is when the cell's not dividing. It's not part of mitosis. These are our stages of mitosis. We wrote on Friday that the stages were PMATC, PMATC. Those are the first letters. P is for prophase, okay? If we take a look at this picture, we can see chromosomes. We can also see spindle fibers. So remember, in interphase, we had messy spaghetti, okay? It was chromatin. If we try to take that messy spaghetti and split it in half, it might get tangled. It's going to be really hard to do. So the chromatin organizes into chromosomes. If you wrote, we have chromosomes, I can see chromosomes, that would be totally fine, okay? So we can see our chromosomes in prophase. Now remember back in interphase, we said each chromosome gets copied, okay? So what we have then, this X picture that you see up here, this X picture, half of that X is one copy, the other half is the other copy. You can think of it as two skeins of yarn, like this is one little thing of yarn, one arm, and this is the other. This is one copy, this is one copy. They make an X, okay? We have a name for that. If we take a look at the picture down here on the bottom, we can see that each half of the X is called a sister chromatid. They're exact copies. I know sisters aren't exact copies, but it's just the word that scientists give to them. So each X, each chromosome is made up of two chromatids and they're copies. What's going to happen is one is going to go to one cell and one is going to go to the other cell. When that cell splits in half, half to one side, half to the other side. Now, remember in interphase, we could see the nucleus. Do we see a nucleus here? Nope. So no nucleus. If you had no nucleus written down, great. Otherwise, you can write nucleus dissolves or disappears, or nuclear membrane disappears. Sometimes in drawings and illustrations, you'll see the nucleus as a dashed line showing it was there and it's disappearing. And then notice the other thing that's labeled here are these spindle fibers. Do you guys remember when we watched that little video clip on Thursday? It was like the little wiggly worm chromosomes and then they pulled apart. Well, chromosomes can move, but they can't move by themselves. They have to be pulled. So a little string called a spindle fiber attaches and then can pull it to one side or the other. Where does the spindle fiber attach on that chromosome? Where is it on the X? Where does that line connect on the X? The center, yeah. The scientists like fancy words. So what happens here is they gave it a fancy word. And they call that fancy word the centromere, which sounds like center. So the spindle fibers are there, and they attach to the center of the chromosome. And they're going to pull one half of that X to one side and one half of that X to the other side. Each of the sister chromatids to each side. This is the last bullet point in this one. I know it's a lot to fit in this little box. I'll make it bigger for next year. We can go ahead and we can see that this cell here, is in prophase. Perfect. Okay, so let's move on to metaphase. So metaphase is the next one. Our X's, our chromosomes, where are they? They're in the middle, yeah. So meta and middle both start with the letter M. That's an easy way to remember. So during metaphase, the chromosomes line up at the middle or the center of the cell. All right, so here we have an arrow in metaphase, showing it in metaphase, right here. So we need to find another cell in metaphase, okay? So we're gonna find another cell that has the chromosomes lined up in the middle. Now they can't be pulling apart yet. They have to be lined up in a straight line down the middle. Yep, they can't be pulling apart yet. All right, good, there we go. Perfect. So these are some good examples here of metaphase because they're lined up in the middle, okay? And there's technically this one above the arrow could be one as well. All right, next up, anaphase. PMATC, PMAT. This is the A. What's happening to my X's? Pulling apart, yeah. So remember, we said each half of that X is called a sister chromatid. So each sister chromatid, each copy, the half of the X is pulled to each end of the cell. All right, so here I have an arrow pointing to a cell in anaphase where the chromosomes are starting to pull apart. Can you find another example where they're starting to pull apart? All right, so here are our examples. We can see them starting to pull apart. All right, then we have telophase or telophase. You can pronounce it either way. I typically say telophase. Dictionary.com tells me it's pronounced telophase. I don't know. You can call it whatever you want. In this part, notice we can see our nucleus again and our cells starting to pinch and split into two. So I got 2 bullet points for you here. The cell pinches in and the nucleus reforms. I'm not going to have you circle one of these because they're really hard to find. I do have an example to show you though. So this cell, we can see they're kind of pulled to either side and there's kind of a line in the middle. This is an example of one, but there's only really one example in this picture. All right, the last part, cytokinesis. What part of the cell does cytokinesis sound like? The beginning. Cytoplasm. So remember, all of our little parts of our cell need to be split into two. They have to be copied, and then they each have to go to either side of the cell. So like ribosomes, ER, Golgi body. But our cytoplasm, the jelly-like stuff, has to be split into two as well. Technically, this is part of what happens during telophase, but I have it separate. So we're just gonna say here that the cell and the cytoplasm, because cyto refers to cytoplasm, split into two.